Thank you everyone for joining today. For those of you who are watching live and will be watching later. For those of you who don't know me yet, I'm Alex Lemens, Director of our Molecular and Genomics Informatics Core. We also have on the team from PacBio who will be presenting about their awesome new Revio and Onso technology, which we all are excited to learn about. But the general housekeeping items, if you have any questions, drop it in the chat or save it for the end. We will have a kind of a Q&A session at the end. But we can, we'll monitor it, answer things that we can on the fly. Anything else we'll save to the end. But with that, I'll just pass it over to the PacBio team to learn more about their new technologies.
Oh, thank you everybody for joining, and thank you, Alex, for the gracious invitation. We're lucky enough to have Aaron Wenger. He's Director of Product Marketing, speak about the Revio. And then we have Young Kim. He's a Senior Staff Product Manager, talk about the Onso. So gentlemen, I'll let you introduce yourselves and take it away.
Great. Yeah, thanks, Alex, and thanks, Bill. Excited to tell you guys about the new products we're working on. So I think I'll start by going over the Revio, as Bill said, which is our new tool for our HiFi sequencing technology, and then Young will go through the Onso, which takes advantage of PacBio sequencing by binding technology. And at a kind of high level, if you think of the different sequencing technologies and what they're capable of, you can break down two key dimensions: the accuracy of the reads that are produced, and then the length of the reads that are produced. If you look at something like Illumina sequencing by synthesis technology, it'll produce reads that are around Q30 accuracy and 2 by 150 base pairs, so 300 base pairs. HiFi sequencing technology is produces reads that are at the same accuracy or even slightly more accurate at the read level than sequencing by synthesis, but is 50 times longer. So you're looking at 15,000 base pair reads, 15,000 or longer that are at Q30 accuracy. So it really pushes on that length dimension while maintaining high accuracy. And then sequencing by binding is a short read sequencing technology, but it pushes an order of magnitude in the accuracy dimension. So you'll get reads that are also in the hundreds of bases long, but now are at Q40 accuracy, or 1 error per 100,000 bases. So these technologies take of the two different core technologies that PacBio has: the HiFi long read sequencing technology, which uses what's called single molecule real-time sequencing on semiconductor chips. So you get individual native DNA molecules that are affixed to the bottom of these really small wells on the semiconductor chip, and the polymerase sequences a complementary strand to the native DNA molecule, and it does it. The molecules are turned into libraries with circular adapters, so you actually end up synthesizing the molecule many times in series, going around and around the circle, and all those different observations are combined to produce the single read, at what we call a HiFi read. So you'd have a 15,000 base pair molecule in here, a true single native DNA molecule that is then sequenced, say 5-10 times sequentially, and then processed by the system to output a single read representing the accurate sequence of that molecule. The sequencing by binding technology is similar to sequencing by synthesis in that you have a cluster of molecules that are formed, but the key distinction with the sequencing by binding is that it separates the interrogation stage from the incorporation stage. So you first will bind a nucleotide, and that is used to observe the sequence, and then that fluorescently labeled molecule is cleaved, and you incorporate a molecule without the fluorescent tag, and then extend the cluster. So you end up with something like a native DNA molecules and separating and optimizing these two stages allows us to get that additional order of magnitude of accuracy. The kind of way PacBio looks at these technologies is we don't just provide a single DNA sequencing platform. We aim to provide the kind of right tool for the right application. So we have both this HiFi sequencing and the sequencing by synthesis approach. And for some applications, HiFi sequencing is the right one, where you benefit from having long reads at high accuracy. For some, these applications are things like plant and animal sciences, where you'd want to do genome assembly, or immunology, where you want to get entire haplotypes in phase with each other in single molecules. Well, for sequencing by synthesis, that is, sequencing by binding, that excels at pulling needles out of haystacks. So finding molecules that are, reading individual molecules at high accuracies of things like early stage cancer screening or non-invasive prenatal testing. So first, to talk about the HiFi sequencing platform, which is available on our new Revio systems. This is a picture of a Revio. You can see it's about as tall as a person, and this is the work deck with which you interact to load the consumables. The Revio system provides, as its output, HiFi reads, which I mentioned are both long and accurate. But there's actually some other key attributes of the technology. So the reads are long; they're tens of kilobases long. They're at that Q30 or higher accuracy. Importantly, the remaining errors are non-systematic, so they wash out with coverage. The HiFi sequencing also provides uniform coverage of the genome. So there's very good consistency across GC bins. So both high GC and low GC regions are sequenced well. Long tandem repeats are sequenced well, and you get quite uniform coverage of genomes. We're also sequencing native DNA molecules, and that means that you can see things like epigenetic modification. So methylation that might be present in the genome is actually directly present and observable on the sequencer as well. So the HiFi reads, they look something like this. This is just showing a locus of the human genome, a pharmacologically important one, the CYP2D6 locus, and you see on the top HiFi reads and on the bottom Illumina reads from the same sample. The kind of general sense to get is that the reads are of similar quality to each other. So you can look, the Illumina reads and the HiFi reads have similar show similar error rates. You see the same variance across the regions where both technologies are able to access. But there are some regions of the genome, like these two regions here, that are very similar to each other. You're not able to confidently place the short reads, but the HiFi reads are able to just read right through them, and you can anchor onto unique regions to the left and right to place them in the genome. And you see another advantage of the HiFi reads, which is that they are long enough to contain multiple variants, so you can phase them into haplotypes. You can see here, these reads on the top all share the same sequence, and these reads on the bottom all share the same. So you can see one of these is the maternal, one of these the paternal allele. So you get the same variants, but they're in phase, and you extend the new reads to the genome. Just to give a sense of scale, that this is an individual HiFi read, so something like 15,000 bases long. These more these long and accurate reads give a more complete view of the genome than short read whole genome sequencing. So you're not just getting SNPs and small indels. You're getting you are getting those SNPs and small indels, but you're also getting them in phase. You can reach into regions of the genome like segmental duplications. You get methylation in that same measurement, and then also larger variants like structural variants are very well detected with HiFi whole genome sequencing. So it is one of the critical benefits of HiFi whole genome sequencing, that it gives a more comprehensive view of the genome than what you'd get out of sequencing the same individual with short reads. And HiFi sequencing has been used to accomplish a number of amazing things. So one was, it was one of the key technologies in the first telomere-to-telomere assembly of a human genome. It's also been used to produce the first single cell catalogs of not just genes, but spliced transcripts and isoforms. Apologies here. Keep it. And then finally, it was used to produce the first directly phased methylomes. So you can see here an example of where now blue is indicating non-methylated and red indicating methylated. And this is showing an individual that has a repeat expansion that's present in one allele that induces methylation downstream of that repeat expansion. And HiFi was used to produce the first directly phased methylomes for hundreds of human genomes. The longreads are also really good at solving challenging problems. So this is one or two examples, I'm sorry, from Jeremy Schmutz at Hudson Alpha. And he has applied HiFi sequencing but look at humans. And here's an example where he found a pathogenic variant in an individual who had been where they had been unable to identify that with other technologies, and found that the pathogenic event in this case was a 4 megabase inversion. And you can see here that this inversion is characterized when you have HiFi reads not just by looking at the edge of the inversion and seeing like a subtle signal. You can actually assemble the genome of the patient in this case, and then compare that to the reference genome. So now you're looking at the scale of tens of megabases, and you can see, oh, this chromosome 7 in the individual has a very large inversion relative to the reference gene. Jeremy's also used HiFi sequencing to look at complex plant genomes, like the sugarcane genome, and been able to produce both more contiguous and more complete genomes. So you see, when you go to a HiFi genome, you're actually assembling a lot more genome sequence into much bigger pieces. And for sugarcane, that let them find 11,000 more genes in the HiFi assembly than in the starting Illumina assembly. So one of the challenges of applying, so HiFi sequencing is a great technology. It's has been used successfully for very many things, but one of the disadvantages of it has been the scale. So we first launched the system in 2011. At that point, it wasn't even HiFi sequencing; it was just a lower accuracy long-read sequencing. And since that time, we've scaled the throughput 1,300-fold, including this year. Critically, we'd make a 15x jump from what was available on the Sequel II system to what's available on the new system, the Revio system. The key specs of the Revio are that it runs 100 million ZMWs per run. So those like small wells where you're observing a single molecule, there's 100 million of those per run on the Revio. The typical run in a Revio will produce 360 gigabases of data in a 24-hour sequencing run for libraries that are 15 to 18,000 bases long. And with these libraries, you get 5-methylcytosine DNA methylation and extremely accurate bases. So you get 90% of bases Q30 or above. So this is more accurate than what you get on a short read library off of NovaSeq. The kind of key consumable difference for the Revio is a new smart cell, and the smart cell is pictured here. It's a 25 million ZMW smart cell. It shares many things with the prior generation PacBio consumables. So it's a similar chip size, and it uses a similar manufacturing process. So we can produce them reliably and take advantage of the lessons from producing the smart cell 8M to producing the smart cell 25M. But there are some key differences. So one of those is that although it's the same physical size, you're fitting 3 times the number of observations on that same chip. It also has it's our first smart cell that uses a flow cell design. So it has this cap over it and these small ports of access, and liquids are pipetted in through these ports and flow in a controlled fashion over the cell. This improves the loading of the smart cells. We've also improved the illumination uniformity. The flow cell design gives reduced reagent volumes. And then prior PacBio sequencers required nitrogen to keep oxygen away from the surface of the chip. But with this flow cell design, you no longer need an external nitrogen supply. So you just the chip protects itself from the air with this flow cell. So I mentioned it was a 100 million ZMW instrument with a 25 million ZMW consumable. And this is because the Revio is able to run 4 different smart cells at a time. So you can think of these as independent lanes. They're physically separated from each other. There's 4 different observation stages in the heart of the instrument. All of these smart cells can be run independently. They get different samples on them. So you could run something like RNA sequencing on one smart cell, sequence a tumor genome on another, sequence a normal genome on a third, all different libraries. You could also set up one and come back later and set up the other 3. They are largely independently addressable. They do share a work deck and a robot, but down below in the observation, they're completely independent of each other. So we've been running this system now for many months at PacBio. We ran it for many months before we started shipping to customers. Ran a variety of samples in-house, human genomes, mouse, or some animal genomes, and some plant genomes. I think people sometimes ask, why did you run a ladybug and mouse, or why oak and mistletoe? And this is from a collaboration with the Sanger Institute, the Tree of Life program there, they have some standard reference samples, and mouse and ladybug are two of those, and oak and mistletoe are two of the reference plants. So we've ran all these samples in-house, see that you get excellent yields across a variety of different sample types, hitting the quality spec, hitting the read length spec, and the yield spec. So across these runs, and this is fairly consistent with what we've continued to see, you get here over this 90 gigabase yield target, and more than 90% of bases Q30 and above. So it's not just the kind of yields that perform well on Revio. We see that the Revio. Here you can see qualitatively some data from the Revio. This is a human genome. This is HG002, one of the genome in a bottle reference samples, and we're looking at a single gene. So this is a window that's a little over 10,000 bases long. You can see that the reads are very accurate. You can put them in haplotypes, just like the other example I showed before. And then this is an interesting gene to look at because it's an imprinted gene. So it's always the paternally inherited allele is the one that's expressed. That's actually the name of this gene, paternally expressed gene number 3. So you always express the copy inherited from the father and silence the copy inherited from the mother. And that's seen through methylation. So you can see here at the top, the reads are grouped by their sequence. And then the bottom, you overlay the methylation. Here, the red is methylated and the blue is unmethylated. So you can see from this, the bottom copy is the paternal one, the one that's where it's not methylated, it's active and transcribed. So the reads and the methylation accuracy look good on the Revio. You can also see more quantitatively if you look at F1 scores for variant calling for single nucleotide variants, indels, and structural variants. You see excellent performance for the Revio, 99.95% for single nucleotide variants, 99.44 for indels, and 95.59 for SVs. So kind of matching or exceeding where the previous PacBio system had been, and that was the kind of most accurate variant calls you could get. So continue to do that just at larger scale. I'll show a few other examples of data off the Revio. And I should say, please feel free to ask questions along the way, or we'll be happy to take them at the end. But we've run together with the Human Pangenome Reference Consortium some longer libraries. So I think in our spec, we recommend 15 to 18 KB libraries, but the system is capable of running longer libraries. Here in this case, we had a 20,000 base pair library from Human Pangenome Reference Consortium where they made the library, and it was sequenced at PacBio prior to commercial release. This smart cell yielded 118 gigabases of data. You see this is the read length distribution. So the average of the reads was just about what they expected from the library. You got Q31 median read quality, and 93% of the bases were Q30 and above. The genome or the data also assembled extremely well. So the Human Pangenome Reference Consortium is, of course, interested in producing pangenome, producing assembled genomes. And from this single smart cell data, you could assemble the 2 haplotypes of HG002 to complete 3 gigabase human genomes with N50s that are chromosome arm length, so tens of megabases of context size. We've also had customers who sequenced non-human data. So with Corteva, we sequenced a corn line, a maize B73 line. It produced 71 gigabases of data, and they assembled this into what they said was the best maize assembly they'd seen to date out of 160 maize genomes. So they've largely been sequencing on the Sequel II, and saw with a single Revio smart cell they can produce genomes that are complete for corn and are extremely contiguous. So here the N50s was 162.5 megabases. The whole maize genome, which I think is 10 chromosomes, but so something on that order, was assembled into 59 contexts. For a kind of more unique plant, we sequenced the American persimmon, which it's a hexaploid, a large hexaploid genome. The maize genome that we sequenced was a diploid like human, but this is a hexaploid genome. It was sequenced across 3 Revio smart cells to get 267 gigabases of data. you can look at the data is accurate enough that you can do K-mer level statistics to estimate the ploidy, and you can see in these genome scope plots these multiple peaks, which is the counts of the K-mers. You can see from the number of peaks you can estimate that it's a hexaploid, and then it was assembled into a very complete or a very contiguous genome. For the persimmon, we don't have a reference like we do for maize or human to know what the expectation is. But the genome size was about what was expected: 2.1 gigabase haplotypes, 12.7 megabase contig N50, and then from comparing to short read sequencing, they can estimate that the error rate of the assembly was something like 1 in 200,000 bases. So extremely accurate assemblies, even for these non-model, non-animal genomes. As one last example, we sequenced on the Revio some metagenomics samples. This is a metagenomics reference strain from Zymo, the Zymo Biomics fecal reference. It was sequenced on 2 smart cells. Got around, you can see quite similar performance for the 2 smart cells, around 68 gigabases of yield with around 8.5 to 9 million reads. The yield here is lower than the 90 gigabases because these reads are short. So the Zymo reference that the molecules are around this 7.7 kb size. So that's the length of reads that we get. But the kind of counterpoint to that is because the reads are short, you get more circular passes of them and get extremely accurate reads. So these are averaging Q46 in these 2 runs. So yeah, kind of across this wide range of different samples, we've seen good performance, and the Revio system is largely designed to allow people to run at bigger scale and to think bigger with PacBio projects and actually be able to execute those both affordably and practically. And we've seen a few customers who will highlight here who are looking to do much bigger projects on PacBio by taking advantage of the Revio. So MBRU in Dubai has purchased multiple Revio systems for many thousand human genome projects focused on genomic medicine in that country. The Wellcome Sanger Institute has purchased multiple Revio systems to ramp their Darwin Tree of Life program, which is aiming to sequence the genome of every organism on the British Isles. They're also looking to increase long reads in human applications. And then Radboud University Medical Center in the Netherlands, they've been using HiFi sequencing for a few years to look at rare disease cases, and they're going to be moving from the hundreds of human genomes they've been able to do now to thousands of genomes with Revio. So most of the data that I showed here was generated at PacBio during the development of the Revio system, but we did start shipping them to customers in March. So if you follow PacBio's Twitter or just general genomics interest groups, hopefully you've seen a bunch of the Revio systems appearing around the world. So here's a crate in Sweden. This is one being installed in Maryland. This is one in the UK. And this is one at Hudson Alpha in Alabama. So the Revio systems are now out in customers' hands, and we've started to see the first data coming along as well. So this is just 3 examples. I think these are all human sequencing. So this is from the University of Washington. They did their first Revio run and got 102 gigabases of yield with 18.8 kb read lengths. This is from Radboud in the Netherlands. Alex Hoishin reports that they got 113.8 gigs on their first run with, again, around 17.7 kilobases. And he was competing with Adam Ramir in Sweden. And Adam was a little bit below Alex, but they still got 102 gigabases of yield, which is great, and 14.9 kb read lengths. So we're starting to see the Revios out and performing well. So one last thing, and then I'll turn it over to Young. Just wanted to talk a bit about, I think maybe at your institute, you guys have more experience using Oxford Nanopore long read sequencing than the PacBio. I think when I showed this plot earlier, it was sequencing by synthesis, and then HiFi reads being similar accuracy and much longer, and sequencing by binding just being much more accurate. So Oxford Nanopore produces reads that are a bit longer than the HiFi reads. Depends on the mode. They can be of the same size, or the longest reads for Oxford Nanopore certainly are much longer than the longest reads from HiFi. But HiFi is much more accurate in general. Just wanted to show some comparisons to to both the ONT simplex and duplex for how HiFi would look. So one of those is it showed, just talk about 3 dimensions quickly. First is accuracy. The variant calling with PacBio HiFi, it is significantly more accurate than even the most accurate mode for nanopore. So this is some quite recent data, maybe from last month, or I guess now 2 months ago, February, looking at the HG002 genome in a bottle sample. So this is the sample where we kind of know what the right answer is using the most recent reference for that sample. And actually, these are data pulled from the deep variant version 1.5 release. So the kind of stats that they're all cited here, they're just from the deep variant different case studies. So they had run analysis on 64x simplex data, 80x of Oxford Nanopore duplex data, and then it can compare to 33x of HiFi data. So this is from a Revio system, what you get off a single run. And then the different bins to show the error types that you see. So the duplex nanopore data, which is their more accurate read mode, does give more accurate variant calling than the simplex. This is just for chromosome 20. So if you look at the scale of errors, it's just for one chromosome. And that's included because deep variant does not use chromosome 20 in their training. So it's the most unbiased chromosome to do the evaluation on. So you see that the duplex data does have more errors, but HiFi data has 10 times fewer errors than that. In particular, the indel false positives and false negatives are significantly less with the HiFi data. And that's really the like accurate reads and the lack of the unbiased remaining errors too. So HiFi continues to be the most accurate view you can get of a genome. The other consideration is with the Revio system. If you look at the amount of data you get off a Revio cell, so it's this 90 gigabases per flow cell, or per smart cell as we call them. If you compare to the current yields on the PromethION flow cells for duplex, they would say in a run you can get 21 gigabases off a flow cell in a 72-hour run. So if you scale that back to per same unit time, you're looking at 90 gigabases off a Revio flow cell versus 7 gigabases off an ONT one. There are other important differences about the system, which the Revio can run up to 4 flow cells at a time, so 360 gigabases in a day. The nanopore systems can run, depending on the system, up to 48. So you'd see that a Revio system running at its full capacity, which is relatively easy to do, loading the consumables, would produce similar but more data than a nanopore system if you were able to load and run all 48 flow cells at a time. The last piece of that is just on the cost. If you look at cost today on a Revio system, the Revio system itself, and I'll show the price in a second. The Revio system itself costs $779,000, and the consumables cost $995 a run. If you look at running that at typical utilizations, this is not at like full utilization. You see the contribution for Revio of a consumable per gigabase is around $11, and the instrument amortization is around $2. For the nanopore systems, the instruments are cheaper, so the instrument contribution is smaller, but because the Revio consumable, which is similarly priced to these, produces so much more data, the per gigabase costs on the Revio are less than on these other systems. And I think I'll end by talking about the price of Revio, and then happy to take any questions on it. But so the key specs of Revio system, it produces 360 gigabases of HiFi yield per run in a 24-hour sequencing time. It can do 1,300 human genomes per year to 30x coverage. Of course, it can also do other applications. The list price for the system is $779,000, and the consumables for the run, or per genome, it ends up, the unit is relatively easy because about one flow cell is 90 gigabases is 130x human genome, and those consumables are $995 list price per run. So you can think $995 human genome, and up to 4 of those a day. So that was all I had on the long read side. I don't know, Alex, would you like to do questions on this now, or shall we move over to Young?
I'd say let's move on to Young, and we could do all the questions at the end.
Okay, perfect.
Perfect, thanks.
With that, show is yours, Young.
Okay, sounds good. Thanks, Aaron, for the overview of the Revio. Let me share my screen. Okay, perfect. All right, well, thanks so much. We're going to go to the second part of the talk and talk a little bit about the latest addition to the PacBio family, which is the Onso system. And this is PacBio's first ever short read platform. And this is obtained through our acquisition of a company called Omnium back a year and a half ago. And we're trying to build out sort of the story here as sort of the most accurate short read system available in the marketplace right now. So as of right now, Onso system is not available commercially, but we intend to commercialize and have our first customer ship at first half of this year. So let's talk a little bit about the key features for the Onso system. Again, it's really about the accuracy for the Onso system, because right now in the current market dynamics, it's usually dominated by Illumina short read systems, MGI, Element, Singular, and a host of other players in the short read space. And a lot of times they focus on sort of throughput and cost per gig, but the accuracy has not really changed over many years. And so this is an opportunity for us to innovate and potentially drive many types of applications where accuracy does matter. And so Onso right now is really a mid-range sequencer from a specs perspective. So I think it's similar to any NextSeq or some of the other mid-range sequences that are currently in the marketplace. And so we're offering around 400, 500 million reads per a single flow cell. And we are planning to commercialize a 200 cycle kit and a 300 cycle kit at launch. And then like I said, the accuracy is really the standout in the story here. We're expecting greater than 90% of all total bases are at Q40 or more in terms of the system. So that's a log higher than some of the other specs you're seeing from the short read players. And the last point is that we're offering also a library prep, a complete library prep solution tuned for Onso's accuracy, as well as a set of kits that will enable conversion of existing Illumina type of libraries to SBB or Onso specific libraries, so you can directly compare your Illumina data with SBB data to understand what the changes are. So for Onso, as right now, it is currently in an external beta program. And so we have 3 institutions. These are well-known institutions, obviously, that are sort of putting Onso through the paces. And an initial glance right now is that from running the samples and doing the testing, and many of them been doing since November last year, is that the data has been really favorable, and it really sort of speaks to what we've been saying internally in terms of the quality of data and the accuracy. And so they've been really pleased with the results of to date. And just to kind of talking about data here, so for Weill Cornell, the Onso is in Chris Mason's lab there, and he ran the first genome in Novato HD002 sample with the updated, most up-to-date chemistry, and he's been seeing Q scores above 50, in fact 55, which is over 1 in 100,000 in terms of error rate. And so he's really been excited, and his whole lab is excited really to test Onso with different types of samples and really sort of prove out the message about accuracy. So let's talk a little bit about what is the secret sauce behind the accuracy, and it is the sequencing by binding chemistry that drives highly accurate reads. And so I think the major component of this is how we separate the binding and extension steps for the short read, which really allows us to fully optimize the chemistry and drive towards Q40 base or higher. So let me go over sort of the steps involved in this whole process. So the first step is from the previous incorporation event, you start off with a terminated nucleotide that's blocked, so you cannot extend to the next base over. And then there's an interrogation step where sets of nucleotides, purines and pyrimidines that are differentially labeled in red and green, are flowed through with the polymerase that are binding next to the site. Again, just on the animation perspective, nucleotides are flowed in, washed to remove unbound nucleotides, and then the image occurs in that step. And then you come up with another wash. And then that's one set, and there's a second set of nucleotides, as I mentioned, that comes in and repeats the steps. So it's two separate steps of nucleotide incorporation, wash, and imaging. And then the third step is activation. So once the interrogation step is completed, the activation step, then what it does is it removes the terminator from the base that was previously there, and then allows the incorporation to happen. And the incorporation is actually done with the same blocked nucleotides as we started off in the first image. So that's kind of the nutshell for the SBB step. And you can see that the two optimized steps here, or separation of the interrogation and the incorporation step, allows control the chemistry that really allows for high signal-to-noise ratio, which is going to be reflected in the Q scores that enables accuracy. And then the last step you can see for incorporation, with standard SBS chemistries, what happens is you have a very large hydrophobic moiety that still remains after cleavage of the dye itself. And so what happens is it accumulates what's called molecular scars, and over a course of sequencing, these things impact the polymerase ability to interact with the strand and potentially lead to mismatch events. And so taking both together, I think those are the key points that drives accuracy for SBB. So you can kind of see from an IGV plot here what I mean by the accuracy and how that differs between SBB and SBS. And you can see here's a region just from CHM13 that you can see on the top with SBS. There's really very low quality reads, and also a lot of false positives in the data, right? That just kind of speaks to some of the issues in the chemistry and the sequencing. But if you look at the bottom part with SBB, look at how clean it is, right? It's basically near perfect in terms of the sequencing. And then this is a region that has a let me have 1 second point this out for you. This is a region where there's a homopolymer stretch as well. And I'm going to talk a little bit about this later, but while SBS does resolve some of it, a lot of it is you have some errors in the after the read through of the homopolymer that speaks to the post-homopolymeric error mode that SBS has. But SBB is not impacted by that. In fact, there's able to resolve a deletion, one base pair deletion in the homopolymer region as well. So in terms of Onso, I think the idea for this is to really drive a plug and play like experience for existing short read NGS customers. So basically what we're trying to do here is to develop a comprehensive library prep solution, sequencing solution that enables existing short read customers to kind of really plug and play into the system. So basically a box swap or a reagent swap, and then you're good to go, right? And the point here is that we don't really want to make adoption a hurdle for existing customers, right? And you just want to basically make this as simple as possible to adopt the box and make sure that the protocols and the sequencing itself is about as close to the market leader, right? So that there's very little learning curve involved. And I think that's what we're trying to achieve here with Onso, albeit the accuracy is obviously much higher. And so we're offering a library prep solution, sequencing, and then a primary analysis as part of the package. And then it should be compatible with anything upstream in terms of sample prep, automation, which we're kind of working on right now in terms of in terms of optimizing that. And then for downstream part with secondary analysis and tertiary analysis, since the output of the machine is FastQ, you should be able to do variant calling and also tertiary analysis as necessary to get the results. So talking a little bit about the library prep, again, we realize there are a lot of already existing short read library prep solutions that are out there, and everybody knows who they are and they have their favorites. But the library prep that we are intending to launch with Onso is actually, there's a value add to that. And it's really about the accuracy. And so we found that when we started off in the development process, our original library prep has some biases in terms of read errors. And so there were some issues with mismatches in terms of doing the library prep, where one read has had more mismatches and therefore drops in Q-score relative to the other read. And so we identified the issues there and developed a solution where now both of the reads have very similar Q-scores across the board, and in fact these Q-scores are now hitting over 50, right? And so this basically from the library prep side, removes a lot of the sort of upstream library prep errors that are inherent to existing short read library preps that are out there, and then it really sort of is in tune with the accuracy of the native accuracy of the Onso sequencing itself. So we believe that pairing up PacBio's library prep with the Onso is likely going to generate the most highest quality data that short read sequencing has ever seen. And so on top of that, as I mentioned earlier, we were selling index adapter plates with very, very low index hopping rates, less than 1%, and as well as a library conversion kit that will allow conversion and comparison with existing SBS data. So let's talk about some of the data that we've generated in-house for the SBB platform. And again, as Aaron alluded to earlier, talking with Onso, it's really the sort of the needle and haystack type of applications that we believe is the most suited for this particular platform. And in this particular slide, what we did was we're looking using basically analytical standards. Some of you guys might be familiar with this with Sericare. ctDNA mutation mix is an analytical panel with a mix of different variant allele fractions, and then using an off-the-shelf library prep from Agilent, the XTH2 kit, and their enrichment panel, target enrichment panel, comprehensive cancer panel covering around 150 genes. And then we took that material and then actually converted it to an Onso compatible library using the library conversion kit that I mentioned earlier. So as you can see here, at any variant allele fraction below on the x-axis, and then the sensitivities on the y-axis, it's that at equivalent depth, 6,000x coverage, you can see that the sensitivity of Onso is much better than SBS, and you can see that across the board until you hit the latter points. Let me put this back here. And indeed, actually, when you do these types of experiments, typically for SBS, a lot of users put in UMIs to error correct for any PCR errors, as well as potentially library prep errors that can occur due to events like cycling at high temperatures over time causes abasic sites and things like that. So you have to use UMIs to correct for these types of events. But in this particular case, we still didn't use UMIs, so we kept that at 6,000x with no UMIs for SBB, and compare that with fourfold more depth of sequencing on SBS with UMI, and you can still see that we're outperforming in terms of sensitivity across the board with at different variant allele fractions. And so we've actually internally seen when you go even higher with SBS with UMI, 48,000x, 60,000x, and 80,000x, there's very little return on in terms of their sensitivity. In fact, actually, the sensitivity keeps going down because at higher and higher sequencing depth, even with UMIs, you're injecting more and more and more errors. And so therefore you're capped in terms of what you're able to achieve. And so we believe that, a highly accurate Q40 platform indeed can outperform something that's sequencing a much higher depth, and therefore potentially help improve economies of scale in terms of cost and number of samples that can be run. So we're going to switch gears a little bit and talk a little bit about some of the other advantages of SBB that we identified in sort of R&D, and also some of our and customers who've run some samples on these. And so one thing that we've noticed that is the performance of SBB is really, really great in regions with homopolymers, so poly A, T, CGs. And so you can see here in this graph that on the x-axis is the length of the homopolymer in question, and then the y-axis is a fraction of the correct what fraction that is known to be correct length and identified by SBS and SBB. And you can see that for PacBio SBB, it's regardless of the length, it's very, very flat in terms of the fraction correct length, and there's a dip here. But you can see that with SBS, due to their issues with post-homopolymer errors, increase in homopolymer length is results in decreased number of actual accurate correct lengths. And so this is where we see that homopolymer enumeration is something that we do really, really well versus SBS. I'll show you some examples of this. So this is a TOM40 locus, which is actually a clinically relevant locus involved in Parkinson's disease and such. And this is a poly-T tract that is located in this particular region here in red. And while with SBS, you can see that the quality of the reads are low, and some of the poly-T region is not fully resolved yet. While we can see with SBB that not only is it resolved, but it's a heterozygous condition, right? And so you have half of it's basically a shorter T versus a longer T, which has clinical implications in terms of disease management going forward. So this is a good example of how it's able to be sensitive enough to read through these regions and distinguish a hetero from a homozygous. Another good example here is a poly-C region here. And this is actually in CHM13. So this is actually a control sample where you wouldn't see this poly-C, the pathogenic variant of it. So the wild type is 7 Cs, but then the pathogenic is 8 Cs. And this is, I believe, a disorder we'll call kidney polycystic kidney disease, where you have the extra C that leads to a disease phenotype. And you can see here that with SBS, because of the post-homopolymeric error mode, you can see a lot of these are actually false positives. So you're actually tacking on another C to it from what is expected to be just 7 Cs. And with PacBio SBB, you clearly see that it's still 7 Cs. But with the extra C here, this control sample now becomes potentially pathogenic. And so I think this is one of those examples where the false positive for SBS could be of importance, right? You can make the wrong call, which is not a good thing, obviously. So this is where we believe we can excel from that perspective. And again, another good example here is like really reaching the limits of sensitivity for us, right? Like, so basically how far can we go and without even, doing UMIs or error correction, right? And trying to see, what is the point where we're continuously linear, even if we dilute samples down to a particularly low allele fraction. And so you can see here, this is an amplicon, and this is done with a collaborator for us, where they developed a Mycobacterium tuberculosis amplicon, a gene that is actually involved in resistance to MTB drugs. And they want to kind of identify like early stage events and mutations that can drive resistance. And so here's what they did. So they PCR'd up an amplicon in that region with the mutation there, and they basically said, okay, well, let's go from 10% mutation all the way down to 0.001%. And so basically as a synthetic dilution series of the amplicon. And you can see that even at 0.001, you're able to pick up the mutant counts, right, amongst the background of wild type, right? And really demonstrates that, with SBB, you can really challenge the system to get down to the very lowest level of sensitivity that's possible in a particular sequencer. So this is one good example of what we can bring to the table with SBB. And switching a little bit, so again, we've been talking about the accuracy perspective and how SBB can deliver on that perspective. But just to remind you guys that also this is a mid-range sequencer in itself, so it can do other applications that a mid-range sequencer is enabled to do. And so this is in collaboration with 10x Genomics, and basically, we took their 3' single cell library and converted to an Onso format. And the results here are pretty striking. In fact, you can see that on the Q30 on the highlighted box here, Q30 of the RNA read is actually 99.8%, which is actually unbelievable, which is something I've personally never seen before. But it's highly accurate read through this particular library. And then when you look to the right and run it through Cell Ranger and identify the fraction of the different T cells and monocytes and populations on this PBMC sample, these correlate really well with existing data sets that were generated from SBS. And so this is really a good example that we're more than a needle in a haystack box, and it could support other types of applications like single cell as well. And internally, again, this is not in commercial stage, and we're hoping to commercialize in the next few months. But I think some of the runs that we've gotten internally have really exceeded our expectations, especially with the Q score. So we're trying to hit 40% or 90% or greater at Q40, but we're seeing higher than that, like 46, 44 for some of the runs, and read lengths of 2x150 are achieved. And then sequencing time around 48 hours. And so we're still working on things like, flow cell cluster density, but we don't believe that's going to be a showstopper for us. And I think that we'll be well on our way to hitting around 800 million to 1 billion paired end reads by the time we're at launch. Actually, this is just internal data, but the data from our external beta sites actually exceed these in terms of the number of reads. They're comfortably hitting around 800, 850, almost 900 in many of the sites. So in real world use, actually, I think they're already meeting and exceeding the commercial specs that we laid out. So a little bit about the Onso consumable pricing. And so for the 300 cycle kit, it's a shade under $2,000, around $15 a gig for 120 to 150 gigabases of output per kit. There's also a 200 cycle kit that's at $16.95 and priced a little bit higher in terms of price per gig. That's output for 80 to 100 gigabases a kit. And each kit contains the reagent pack that enables the cycling to occur, a clustering reagent plate, as well as the flow cell kit. So this should be fairly standard, if you're in the short read space in terms of your expectations of what you need from a kit like this. And right now it's available for pre-order, and it has been since January at JP Morgan conference, but that's an option for you. So let's talk a little bit about, we said that we've seen the data that shows up to 4-fold less sequencing required for SBB compared to SBS. And so what does that mean in terms of potential savings for labs? And so obviously economics matter a lot for a lot of these labs running these types of tests. And so we have an example here of a panel, we'll just call it panel A and B, that are available commercially, typically 500 genes, gene panel for liquid biopsy testing. And for SBS, they would recommend a target depth of 25,000 to 35,000 X coverage. But for Onso with 4-fold less sequencing, that means you get 4-fold less needs depth to reach a particular variant profile. And so from a gigabase per sample perspective for SBS, we're looking at around approximately 75 gig, and these are all max output as stated by the manufacturer. But for Onso, it's going to be a fourth of that, right? So around 18.8. So what does that mean? So when you look at it from the perspective of sort of the running costs for the flow cells, the kits, any amortization impact in the sequencer, we're landing around $300 versus SBS cost, which is around $700. That's really a fairly substantial savings in terms of a per sample, around 57%. And so if you're running a liquid biopsy lab and having to sequence at these high depths, you may want to consider, putting in a more highly accurate solution to really drive the cost per sample down and improve the unit economics for your lab. So in summary, so here are some of the key sort of benefits of Onso. Really, it's about the accuracy, right? 90% over Q40. That's unheard of. And so, people are going to be really excited to really put Onso through the paces in terms of looking at new applications that can take advantage of the Accurate platform. And then again, compatibility, right? We didn't design this to make it difficult for customers. We wanted to have it as much as plug and play as possible, from the library prep kit to sequencing all the way to the output with the FASTQ file. And then obviously with the accuracy, needle in a haystack application. So it doesn't necessarily have to be even liquid biopsy. Anything where you believe you're trying to look for a variant within a population of wild type is fodder for Onso and the technology behind that. And again, with the last example that I showed you, it's reduced sequence needs versus existing short read sequencers, and that could really help drive costs down for those labs. And here's information on Onso. The list price is $259K, and you can learn more on the QR code here. And it will be available in the first half of this year for full commercial launch. And with that, that concludes my part of the talk. And so I guess we can open up for a Q&A session, I believe.
Mm-hmm. Yeah, awesome. Thank you. Great presentation, both of you.
Thank you.
Very informative. I guess if anybody has any, while everyone's thinking of their questions, I got a couple quick ones I could ask. For starting with the sequencing by binding, which is really cool. I was wondering, I imagine it probably is, but is it in your roadmap to eventually scale it up to those larger capacity sequencers so it can, compete with like the NovaSeq, for example? I'm probably a little bit further down the roadmap, but
yeah, absolutely. Yeah, we're always assessing the market here, right? And obviously, liquid biopsy and those type of ctDNA type of application, there's a lot of sequencing that needs to be done, especially when they're going to scale up to thousands and thousands of samples, right? And so, yes, we definitely have ideas for bigger and faster and larger sort of outputs, right? That's in our roadmap going forward in the future. Yes.
Okay, cool. Then, I guess a question for Aaron with the Revio system. So for I simultaneously hate and love when people say, "Oh, it's a X dollar genome, like sub 1,000 for the genome," because it's always just the reagent costs. So the bioinformatics associated with that, because I imagine it is, it's with these longer reads, it's usually a little bit more computationally heavy. Do you have like an estimated bioinformatics cost for doing the let's say a standard like variant analysis comparison?
First I'll just answer that directly, which is, the number we usually said, if you'd want to run our recommended tools on a cloud, so paying cloud rates, it'd be around $15 a genome.
Okay.
And then if you want to do genome assembly, which is sometimes something people do so that's for structural variant calling, or it's read alignment, structural variant calling, and then small variant calling. And for that, deep variant is the tool we'd recommend for that. That's the $15. It's about $10 more if you want to do genome assembly, which usually takes something like 500 CPU hours on a single 48 core node. So that's that. The other point though about long read being more computationally challenging, that's something where I think HiFi reads really help a lot. So you don't have to do any off instrument base calling. You have reads that are like the same accuracy as other algorithms have been built around, right? So these aren't noisier reads that you have to correct or be a little bit more lax with your parameterization to map. So it is actually pretty efficient. So I'll say two more things. One, all the base calling and stuff happens on these, but you get the HiFi reads directly off. You get the methylation calls directly off the instrument in the read files. And then the last thing, in Revio, we made an attempt to. It's the first time PacBio did like binning of quality values, like you may be familiar with from NovaSeq, right? So you're not representing all 94 possible quality values. On the new system, there's 7 quality values, and the reads are sorted in an intelligent way to like compress well. So the files are actually reasonable sized. It's around, I think our spec is 55 gigabytes per run. So it's usually something like half a byte per base. So 90 gigabase file is something like 45 gigabyte size. So it's actually reasonable sizes. Yeah,
helps with the data storage issues.
Yeah, exactly.
Yeah. And the methylation too. I'm glad you brought that up, because that's really cool to see the methylation data, especially that one that you showed, the PEG3, that was so very clear. Night and day with that. Can it do other methylation calls besides just 5MC?
So I think the system only calls 5MC at CPG sites right now. The technology, smart sequencing, has been demonstrated to be able to call many other types of methylation, but the one the system provides is just the 5MC. There's also a way to get so I didn't talk much about how the methylation actually works. So you may know, which is that there's the sequencing is incorporating fluorescent labeled nucleotides, and the kind of label denotes A, C, G, or T. But then the sequencer is the polymerase is moving in real time at its own cadence, and that second dimension of time, the kinetics of the polymerase, is what's affected by the methylation. So if you have a methylated C, you still are going to incorporate a G complementary to that, and that's going to be the same label as a G complementary to non-methylated C. So there's no signal there, but then how long it takes to do the incorporation, how long it takes to move down to the next strand, that is impacted. So those kinetic values, you can also export those from the system, and then you can run off instrument methylation callers. In practice, the only other one people use is for 6MA and 4MC in microbes. And then there's some neat like biochemistry techniques built around 6MA in human. So I don't know if you've seen, there's this technique called FiberSeq developed by Andrew Sturgakis at the University of Washington, and Vijay Ramani at UCSF developed a similar technique called SAMOSA. And there they use 6MA methyltransferases to add adenine methylation to human genomes, and there's no native adenine methylation in human genomes. So the assay they primarily use it for open chromatin detection. So if you kind of treat samples with this methyltransferase, all the exposed A's will get methylated, and then when you sequence it with PacBio, you'll get a signal at those A's. And you can say, hey, these are my methylated A's. I know that must have been open chromatin, because there's no native biological 6MA. So there are callers for that too that use the kinetics from the Revio system.
Oh, that's cool. That's awesome that they can capture pretty much ATAC data without doing the ATAC-seq assay.
Yeah, it's really neat. I think if you do so, I think you can think of a standard PacBio whole genome run as like bisulfite plus variant calling in assembler, because you get the reads, you get the 5MC. Yeah, and then if you add this, if you do FiberSeq, then you get from the single, it's still the single sequencing run. You'd get the sequence, you get the 5MC, and then you also get the ATAC-seq equivalent. Yeah.
That's cool. That sounds like a lot of fun. Any questions from the audience going on? That's pretty much all my questions. So, yeah. Okay, cool. Well, then I guess thank you everyone for attending, and thank you PacBio team for an awesome presentation. I definitely learned a lot today. This is very interesting. And hopefully we'll have this posted over the next couple of days. But if anybody has any questions, obviously feel free to reach out to myself or anybody in the PacBio team, and we'd be happy to help you as best we can. So thank you, everybody.
Yeah, thanks for joining.
Well, thank you, Alex.
Have a great day.
Thank you, Aaron. Take care now. Bye-bye.